Good morning, folks. This is Mr. Dan coming to you from the Landscape Design Classroom, and today I'm going to talk to you about basics of plant growth. So, ways to measure plant growth: fresh weight, dry weight, volume, length, height, and surface area. So, what makes a plant grow the way it does? Form and shape change with the plant's overall size due to genetic factors. Plant growth occurs because the number of plant cells increase through division, and the size of the cells increase as they become differentiated into specialized tissues and organs. Heredity and environment interact to determine growth and growth patterns. So, where do new cells come from? Tips of stems and roots, that's increasing length. Axils and leaves form new stems, leaves, and flowers. In cambium layer in stem and roots, increasing diameter of stems, roots, and tree trunks. So, Meristem, this is the term applied to a region of plant growth. So these cells actively divide. They also mature, increase in size, and change into specialized tissue. Differentiation changes in cells to form specialized parts of the plant. Some example of this is in a young root, differentiation results in the formation of different types of tissue for conduction of water and food, storage of food, protection, and the uptake of water and minerals. So 3 general cell functions. You have maintenance, synthesis of cell products, and cell division. So all these functions require energy. ATP, adenosine triphosphate, is the universal energy transfer molecule. So morphogenesis. The plants begin as a single cell, but by repeated cell growth and mitosis or division, eventually contain billions of cells. This process of development is called morphogenesis. Morphogenesis involves not only cell growth, but differentiation into specialized types of cells. This differentiation is controlled by the genes. Each cell manufactures the proteins and structures necessary for its function. So, genes are the basic unit of inheritance. Genes are carried on chromosomes in the cell nucleus. Genes contain the blueprint or code for how the plant will look and interact with its environment. The number of chromosomes vary from species to species, but it's consistent with the species. Chromosomes come in pairs in normal cells, and genes are made of DNA, so deoxyribonucleic acid. And DNA is what we are all made of. So DNA is pretty much in anything living. So the genome is the master blueprint, a complete set of instructions for an organism found in every nucleus of the cell. The genome consists of tightly coiled threads of DNA and associated protein molecules, organized into structures called chromosomes. So what about DNA? 2 strands that wrap around each other to resemble a twisted ladder whose sides made of sugar and phosphate molecules, connected by rungs of nitrogen-containing chemicals called bases. And each strand is a linear arrangement of repeating similar units called nucleotides, which are each composed of 1 sugar, 1 phosphate, and a nitrogenous base. So the 4 bases of DNA: adenine, thymine, cytosine, and guanine. So the order of the bases is called the DNA sequence, and each time a cell divides into 2 daughter cells, its full genome is duplicated. So controlled growth. Heredity, the general species characteristics and limits on size and rate of growth. And hormones, nutrition, and environment. The role of plant hormones. Classes include auxins, cytokinins, gibberellins, abscisic acid, and ethylene. Cytokinins are especially important in cell division. Elongation is promoted by auxins and gibberellins. Several hormones interact for cell and organ differentiation. The initiation of roots by auxins and buds by cytokinins depends on the presence of opposing hormones in the proper amounts, and hormones regulate other growth-related activities, including seed germination, flower and fruit development, and leaf enlargement. So the environment plays a part. Temperature: warm temperatures favor more growth, cold is required for some seeds to germinate, some buds to begin growing, and some plants to flower. So light regulates flowering, leaf expansion, stem elongation, movement, and leaves and seed germination. So everything you see in that picture on the right here of this flower and the plant involved, everything comes from different processes of photosynthesis, everything we've been talking about in these last few lectures here. So plant growth regulators, organic compounds other than nutrients that modify plant physiological processes. PGRs act inside plant cells to stimulate or inhibit specific enzymes and enzyme systems and help regulate plant metabolism. Used to modify crop growth rate and growth pattern, may occur naturally or be synthetically produced. So some examples of PGRs. Auxins act to stimulate or inhibit growth, form roots on cuttings, etc. Gibberellins control cell elongation and division in plant shoots. Cytokinins act in cell division, cell enlargement, sustenance, and the transport of amino acids in plants. These three are often used in combination for specific effects. Inhibitors prevent enzymes from breaking down, and ethylene is often used with auxins to regulate metabolism. So I know that was a lot to take in within a short period. If you have any questions at all or need anything at all, give me a call down here in Landscape Design Classroom, and I will gladly answer any other questions you may have. Remember to look back on this while completing your review worksheets, any of your vocabulary, and anything we get into in class, and which we will be expanding on this in the classroom. But if you have any questions at all, please get in touch with me down here in the Landscape Design Classroom. And I appreciate your attention, folks, and have a great day.